we've looked at is called the cortex, and the inner layer is the medulla. And here's where it gets a little different though. So you're going to have to, on the questions, look at what I'm asking. If I want the layer out here, it's cortex. If I want the layer in here, that's medulla. So the layer is what I use. I want those. However, if you look at the medulla, it's set up as a bunch of sort of triangular blocks of tissue. These triangular blocks, they look like a pyramid. They're called renal pyramids. They're easy to see on the preserved kidney. You can see them here. See these dark red triangular bits? So those are the pyramids. So if I point to this, and say identify the structure, what are you going to tell me? Pyramid. If I ask the layer, well, it's the medulla. So the medulla is made up of pyramids. So it's kind of subconform. Does that make sense? You'll have to look at the question. Otherwise, if I stick a pin there and say, what's that? You could say medulla, you could say pyramid. Right?
So if you say layer.
Layer medulla, structure pyramid. Got it?
Okay.
Then, there is some cortical tissue that kind of dips down between the pyramids. See, it's shown pretty well, like right here. See, there's a difference in the texture. The cortex and the pyramids, they have a different texture to them. But you see how there's this big glob of stuff sticking down between these two pyramids? That's called a renal column. It's basically a piece of cortex that dips down between the renal pyramids. So this would represent renal column. Then, the little apex of these pyramids has its own name.
Oh my God.
These little bits right here, the little pointy end of the pyramid. It's called a renal papillae. A papillae means a little bit that's kind of sticking out. So if you look right here on this bottle,
I don't see it.
there's a papillae, there's a papillae. Basically the apex of all the pyramids is a papillae. And the reason those are important is around each of these papillae is a little tiny hollow cup-like structure. So, Gail.
Okay.
Oh.
All right. So these little spaces in front of the papillae, they're actually hollow cavities. There's nothing there in these bottles.
Wait, so they're all like connected? Really?
I'm getting there. These little hollow spaces right here, I'll put some little hash marks on it.
Mm-hmm.
Those are referred to as a minor calyx.
What?
Minor calyx. I didn't name it, don't blame me.
Wait, hold on.
See this little hollow space right here where the pencil tip is? That's a minor calyx. In here, minor calyx. Here, minor calyx. All the little spaces in front of the papillae. You see, here's what happens. The kidneys filter the blood, right? Something like 30% of your cardiac output goes to the kidneys. That's a lot of cardiac output for some pretty small organs. As it passes through, the junk you don't need, we'll call it waste, gets removed. That's what your urine is, right? So it's got a lot of urea in it, up in nitrogen waste, excess water, excess salt. That's processed by structures in the cortex and the renal pyramids. In fact, if you look at the renal pyramids carefully, even in the preserved kidneys, you'll see a bunch of little tiny pinholes at the end of papillae. Those are the drains. As the blood is processed, the waste material is pulled out, and you'll see it dribbling out of these little holes right here at the renal papillae. And they fill up the minor calyx. It's like a little cup sitting under that little thing with the holes in it. And the urine flows in there, fills up the minor calyces here, 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 here. Then, yes, as you noticed, Jennifer, they all run together in this much bigger space. That one's called a major calyx. And so you're watching urine flow. The urine will collect these minor calyces, flow out into this bigger space called the major calyx, then flow out this little funnel-shaped bit, which is called the renal pelvis. And then it goes out into the ureter. So this is your ureter. That's a ureter. It goes down to the bladder and then out. So you got that?
No.
Pyramids, papillae.
Wait, I missed that.
Pyramids, papillae. The urine drains out of the holes at the end of the papillae into the minor calyx. So these minor calyces will then flow together into a bigger space called the major calyx, and then urine will flow out through a little funnel-shaped bit called the pelvis, and then it drains into the ureter, and it's into the bladder and out of the body. So, on this, see these minor calyces here, I put my fingers on them. They all drain into this bigger space here. That's the major calyx, and then it drains out this funnel-shaped bit, that's the pelvis, and this is the ureter that goes down to the bladder.
So you have a pelvis in your kidney.
Yeah, this funnel-shaped bit.
Okay.
And what pelvis means probably, I haven't looked up for a while.
Never knew.
Like you could call it the renal infundibulum if you like.
Oh, please.
No, it's not. That sounds even harder.
So that's kind of the pelvis, just what kind of shapes like a funnel, and then it drains it into the ureter, out to the bladder, through the urethra.
So ureter's
And
like
onto
the
the
very
trees.
end.
Ureter's the tube that's going to go from here down to the bladder. What else? On the large scale, that takes care of papillae, pyramid column,
Is that only gonna be on the model?
That will be on the model and more. But wait, we're not done. All right.
Yeah.
Preserved kidney.
How long is
What animal is it
it?
from?
I like doing how long.
Pig.
So they're big?
If you ever see a cow kidney, they look crazy. Pig kidneys look like ours. Cow kidneys are lumpy on the outside. They're crazy looking things. Pig kidney looks more like ours, just a little bit bigger.
Which is good from? Yeah.
Because we're not like pigs.
Opportunity.
I mean, what do we eat? Whatever we can catch. What do pigs eat? Whatever they can catch. So we're very simple.
Yeah.
They get to eat the scraps. They don't get one
I don't know, they'll eat anything.
Oh,
That's a section pig kidney. See, they kind of look like ours. It's bigger than ours. But,
I thought you said it was lumpy.
no, that's
Oh,
the
yeah.
cow. Excuse me, Jennifer, I want to get some dissecting stuff here.
Oh, God, it stinks.
So on this one, the cortex is about a centimeter thick. All this is cortex. You'll see the pyramids. They have a different color look, different texture. Those are the
Wait.
pyramid. Cortex kind of looks
Why are
like
those things
hamburger
smell
meat.
so bad? Jesus
Pyramid,
Christ.
pyramid.
That's a kidney
See in this little space right
from
here?
a cow?
That's a minor calyx. That little
I think so.
tip there is a
Or
papillae.
pig. I don't know.
Renal columns are
That's a
kind of
big
coming
kidney
down
for a
between the
pig.
pyramids. So
Did you say
all
it's
the
a
same.
cow or a pig?
This is a pig.
Okay.
That's a big kidney.
Cow kidneys are really lumpy. Pigs get 500, 600, 700 pounds.
Oh yeah, true.
They can get pretty big.
I guess because they're just not tall. Maybe
You don't think
I don't think it. I
So
think
what
they like to
we've
be
gone over
smaller.
so far is on the preserved kidney and it's on the model. Now the next stuff is going to be more microscopic. You won't necessarily have to look at it. Well, you won't have to find it on the preserved kidney. So let's just go through the next sections. Let me erase this. And I'll draw it a little different, so now we'll have a bit of cortex here. And I'll put in one pyramid. So this is just the one pyramid that's overlying cortex.
Change my answer.
Now, imagine you could zoom in with this microscopically. You see all the white stuff on this middle box right here? That's what we're gonna look at. The kidney is made up of millions of little microscopic tube-shaped structures, twisted up tubes. They are called
Wait, what's called that?
the twisted up tube things. These little microscopic tube-like structures that make up the bulk of the kidney, like 10 million of them in the
Oh,
kidney.
the thing that looks like a stem.
It just looks all white and wiggly in here.
Yeah.
That's called the nephron. This is a part we'll actually look at in lecture more than the rest of the gross anatomy. The nephron you can think of as a functional unit of the kidney. That is, if you know how one nephron works, that explains how the kidney does. So we'll multiply by 10 million. So there are going to be some different parts of the nephron we have to talk about. And keep in mind, this is not something you can see on the models of the kidneys. They're microscopic. So let me position a nephron in here and we'll look at the parts.
So we won't be labeling this. We won't have to identify this in here, in lab. Or we will?
Yes.
We will?
We
I
will
would say,
it's on that bottle.
what is this? What is this?
Oh,
What is
okay.
this? What is
Okay,
this?
okay.
What is this? Yes,
Gotcha.
you must know this. Or I would keep drawing it on the board.
Okay.
Oh, that looks like a, the little punchy thing. I
It
can't
looks
think of what
like
it's
the
called.
pedicularia of a sea urchin, actually.
No.
Yeah, it does.
I was gonna say like a little crab, like
Oh, yeah.
claw. That's what it's called. A claw. Like a crab.
Oh, I do.
All right, that's my diagram of the nephron. What you'll notice, the nephron starts at a structure that's often described as a double-walled cup. There's an inner wall here and an outer wall here. This is hollow, or it's the lumen. It's actually what you're looking at really enlarged. The white part here, this is part of it. This structure, this beginning of the nephron, is called
So, like, crab man or something?
yeah. You know, it's almost like you punch your fist in a balloon. That's what you got here. Here's your fist. You got a layer of rubber here, air, layer of rubber out here. That's kind of what we're looking at.
So that's like you're drawing that like that same thing up there. You just
That's
like
this right
okay.
here. All right. This is called the Bowman's capsule. I'll try to briefly tell you how it works in a minute here. We'll look at it in a little bit more detailed lecture. This is where the urine is first going to form. I'll show you how in a minute. But the urine is going to end up in this hollow space right here. I shouldn't even call it waste urine at this point. It's actually called filtrate. Much of it you're going to reuse. But
I
all
need
of
to
the
print this
material
out.
that's
I
in your
printed out
blood
half of it in my freaking printer to have.
is going to end up in the lumen of this Bowman's capsule right here. So on the model, this is the Bowman's capsule. Now, there are two layers to it, right? An outer layer and an inner layer. This is called the parietal layer of the Bowman's capsule. This one's called the visceral layer. You'll need to see out of this model because it's kind of an artist's concept. You see this smooth outer bit right here. This is a parietal, just like with the serous membranes, the outer layer is parietal. This is the parietal layer. Then what happens, you see this twisted up yellow stuff right here?
No. Oh, yeah.
That's the visceral layer. There's a lump of capillaries in here I'll talk about shortly. In here, this red stuff, these are glomerular capillaries. So they're actually inside. I'll just draw a simple one. Right here is a capillary. Let's say. It's going to be more complicated than that, but I have drawing limitations. So this visceral layer of the Bowman's capsule right here.
Wait, the visceral is the inner part?
Yes, is physically attached or wrapping around the capillary. And then it folds out, and this is a parietal layer. So out here is a parietal layer of the Bowman's capsule. This is the visceral layer. It's in intimate contact with the capillaries in it. So this one is actually a better picture than mine, but you can see what they've done here.
But that doesn't look like the little crabby shaped thing.
Okay, this is the crabby shaped thing. I'm sorry. There's
Where?
right here. It's just more complicated. I can't draw this. See this yellow right here? That's the visceral layer. This is a
Okay.
parietal layer. It's still a double-walled cup. There's an inner layer, outer layer. Yes, it's more complicated. It's not a smooth surface here. It's this convoluted.
But it
yellow
doesn't
part.
even look like the
Dude.
little cup. Where's the little
Right here.
part?
This part is this part. In here is this part. I just can't draw.
So
Oh,
the
it's all
yellow
the
dude
way
is
in
the
there.
Three-dimensional and twisted up, yes. This would actually be a lot more convoluted than it looks on my pathetic drawing.
No, I see. Okay.
So that's the parietal layer. Then it folds back on itself and it forms this visceral layer, which is intimately wrapping around these capillaries that are inside the glomerulus, or inside the Bowman's capsule. Talk about those in a minute. This is where the filtrate will start forming, right here. It'll just dribble out. I mean, it's a huge amount. Your kidneys produce like 180 liters of filtrate a day, moves from your blood into this space right here. Then if you follow its flow, this filtrate, it's first formed in this Bowman's capsule. It's going to then drain out this tube right here. This tube, which is this one right here, that is referred to as the proximal, because it's closest to the Bowman's capsule, that's proximal. Convoluted, because it's all twisted up, and it's a tubule. So that's this part right here. So on the diagram, if this is a Bowman's capsule, then this tube leading out, this is part of the proximal convoluted tubule. Down here on the lower magnification, you might be able to see it better. Here's a Bowman's capsule. You see this little twisted tube coming out of it right here? That's the proximal convoluted tubule. Bowman's capsule.
So those are tiny.
Oh yeah, they're microscopic. So there's 10 million per kidney. So this is they're not even this big. This isn't large. This is a proximal convoluted tubule. Then, the proximal convoluted tubule leads down into this big, long hairpin loop that goes all the way down almost to the end of the pyramid, and then it comes right back up. This huge, long hairpin loop is referred to as the loop of Henle.
Loop of what?
Loop of Henle, which is number 17. Long hairpin loop goes from the proximal convoluted tubule almost down to the tip of the pyramid, to the papillae in some cases, and then back up. And then it will drain into another section of twisted up tubule referred to as the distal, because it's farthest away from the Bowman's capsule. Convoluted, it's all twisted up tubule. So on this diagram, over here, you have the Bowman's capsule, proximal convoluted tubule, long loop of Henle, come back up, and now I have a distal convoluted tubule that will drain into this larger tube, going back down through the pyramid, which is the collecting duct. That would be this one right here. Oh, be happy, we're just looking at the names today. When we get to lecture, we'll attach a function to all these. Some of them reabsorb re-needed material, some secrete excess waste, some are able to concentrate the urine so you don't produce dilute urine and dehydrate. Oh,
I thought you said this was easier.
7 more days. I got about 1 or 2 for the respiratory, we'll wrap it up. That'll leave me 2 for digestive and 2 for renal, with 1 extra day to do whatever. Bowman's capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule, collecting duct, and it's a collecting duct that actually drains out the papillae. This is where the urine will dribble out, right there. That's the hole in the papillae. It's actually the opening of the collecting duct. So the nephron, this is the model I'll use. You might notice on this kidney, see that white junk down here? You can't identify what that is, it's too small. So parts of the nephron would be on this one or this one. So Bowman's capsule, I pointed there, proximal convoluted here, collecting duct here, distal convoluted tubule, collecting duct. This one.
So,
All this one has is Bowman's capsule,
okay.
parietal and visceral layer, proximal convoluted tubule, and this thing back here is part of the distal convoluted tubule. That bit right there, see how they almost touch? That greenish one. All right, there's only one other thing we gotta do. That is the vasculature, the blood vessels. You ready?
No.
Some of them we know, come on. What's this artery going into the kidney called?
Renal artery.
Hepatic would be liver, so it is the renal artery.
Oh.
All right, so we already got the renal artery.
That's what I said.
Comes off the aorta, right in the abdomen. Now, as we go in, this is going to change its name and branch a bit. So let me do some stick pictures on the board, and then I'll show you on that model.
No, I'm not
All right, so if we look at a whole kidney, kind of a bulbous kidney. And we'll put a pyramid in here. Pyramid in here, something like that. All right, so
What's that?
renal artery.
It's where? Where?
So I'm starting with 24 is your renal artery. So renal artery goes into the kidney. Once you're in the kidney, it will split into smaller branches. These splits are called segmental arteries. So you can see on this bigger kidney here, here's your renal artery coming in. These are segmental arteries. The major split right after it enters the kidney. So there's a couple branches there and there. If you continue to follow those segmental arteries around, there will be a branch off the segmental arteries that kind of goes up between a couple of the pyramids. So what you're seeing on this one, see here's a segmental, and then you got a branch coming up here and running through the renal column between a couple of pyramids. That one is called, number 27, it is interlobar artery. Interlobar. It runs through the renal columns or between a couple of pyramids. Then it will split and circle around the base of the pyramids. And this one is called the arcuate artery. See, once it comes up between the pyramids, it arcs around the back, so that one right there is an arcuate. If you look here, it's slightly enlarged. Here's your interlobar artery, and it arcs over this way. That's the arcuate artery. It's kind of running right between the cortex and the medulla, along the base of the pyramid.
So the medulla has, what are those little round things?
Those are the Bowman's capsules.
Bowman's capsules,
Up in
okay.
the cortex. All right, then radiating off.
Wait.
Arcuate.
Arcuate, yes.
There you go. I couldn't find it.
I'm going to give you two different names for this one. I think your textbook, every once in a while, it shifts names. On this diagram, it is 28. Interlobular. See, interlobar, interlobular. They're different. The other name you might see in your textbook is cortical radiate artery. Because they radiate through the cortex, cortical radiate artery. Now, we're getting to really small blood vessels here, but there's another branch. Very small. I'm going to change the colors. Up in the cortex, there's some little branches you'll see splitting off the side here of this interlobular artery. These are arterioles. This is probably the best image here. See, here's the interlobar.
Okay.
Here's the arcuate. This is the interlobular going up here. And you see these little branches going off to either side?
That go to the capsule?
Well, those, yes, those are arterioles, but in this case,
Wait.
they name them.
The ones running along outside of the interlobar on the side?
Interlobar here
Okay.
is the same as interlobar here.
Oh,
It's a
okay.
different view. So they're running up along the pyramid. Come up the back, arcuate. And then these ones sticking up through or radiating up into the cortex, these are either interlobular or cortical radiate, because they radiate through the cortex. That's what these are. Then you see these little tiny blood vessels going off on either side towards the Bowman's capsule. Those are arterioles, but they're given a specific name. They are referred to as afferent arterioles. What does afferent mean? Going toward. Efferent, going up. Afferent is going to, efferent away. So these are afferent, and I'm going to have to change my scale here. So this would be interlobular, and then sticking off of those, we'll see a bunch of smaller arterioles. These are called
I have focus on
afferent arterioles. Afferent just means going towards. What's it going towards?
The Bowman's capsule.
The Bowman's capsule is here somewhere. So if we look at this one, this is a better view, this enlargement over here. So coming off of that interlobular artery, this is the afferent arteriole. It leads an arteriole. They lead into what? Capillary beds, right? This afferent arteriole goes into this little knot of capillaries inside the Bowman's capsule. This capillary bed is also given a particular name. It's called the glomerulus. That's a capillary bed. So you have an afferent arteriole going in, then you have a capillary bed. I'll just draw it as a bunch of wiggly lines because that's the best I can do.
I got your whole face in this
So
video.
glomerulus, those are capillaries.
You're YouTube famous.
And then what normally drains a capillary bed?
Venue, not
A
venue.
venule, usually. That's not the case here. This is really weird. Coming out of or draining the glomerulus is something called an E.
Oh, Jesus.
Afferent arteriole. This is atypical. Yes, normally a arteriole goes into a capillary bed and a venule drains it. That's not the case here. You have an afferent arteriole going in, an efferent arteriole going out
that drink.
that drains it. And this is actually going to go to another capillary bed elsewhere in the kidney. I'll mention in a bit. But this is a weird setup, and I'm going to tell you the reason why. Normally, remember how I told you the arterioles are the major resistance vessels in the circulatory system? Did that ring a bell? I think it was a question on the exam.
What?
Arterioles are the major resistance vessels, right?
Yeah.
So you get the biggest drop of pressure when you go through the arterioles. Well, if you look at the afferent and the efferent arteriole, the afferent arteriole is relatively large in diameter. The efferent arteriole is smaller, and it's downstream. So it's weird with this capillary bed, most of the resistance is downstream of the capillary, not upstream. Whereas a normal capillary bed will have about 10 millimeters of mercury blood pressure in it. The glomerular capillaries have a blood pressure of about 60 millimeters of mercury. It's huge. And the reason for that is important. That blood pressure is what's going to be squirting the fluid out of your blood into the Bowman's capsule for processing. So that's partly why I have this weird double arterial setup. Afferent arteriole takes the blood to the glomerulus. Efferent arteriole takes it away. No venule. And of course, the visceral layer of the Bowman's capsule, that's the layer that's intimately wrapped around the arterioles. Now this model can be a little bit confusing. Why is half of it red and half of it yellow?
The visceral layer.
Yeah, they've stripped off the visceral layer on your right here just to show you the glomerular capillaries. They're still here on this one, but they're showing you the visceral layer of the Bowman's capsule intact. Does that make sense? Because I mean, normally this visceral layer would cover all these capillaries. They're just not showing it for purposes of demonstration. And so that fluid will then drain into the space between the visceral and parietal layer of the Bowman's capsule and then go down into the proximal convoluted tubule and get processed, and the waste material will drop out of the collecting duct. So the arteries, I'm not even bother going over the veins. They basically have the same name, just moving the opposite direction. Again, you have renal artery, segmental artery, interlobar artery, arcuate artery, interlobular artery. Then you get into the afferent arteriole, glomerulus, efferent arteriole. Now, the last thing that would happen if you follow this out again, the efferent arteriole will then just go to another capillary bed that supplies the kidney's needs. See, this is just for filtering. This isn't supplying the kidney with oxygen or nutrients or anything. It's just to filter blood. The efferent arterioles will go to another capillary bed, which you can see like right out here. There's a bunch of capillaries. Those are just referred to as peritubular capillaries. There are any capillaries in the kidney that, not counting the glomerulus. Two sets of capillaries. Glomerular capillaries, peritubular capillaries are the other type. And you can actually see it in this model. You see the afferent arteriole going into the glomerulus of this Bowman's capsule, then the efferent arteriole coming out and splitting into another capillary bed, and then the veins are reverse of the arteries, same name, same place, all that.
Hilum?
All right, what is a hilum? Hilum, there's one on your lung sheet too.
Oh yeah, because I remember, but I already forgot. And I asked you,
It's
but I
one of
already
those
forgot
silly
what
terms
you said.
that I probably won't ask, at least not with a picture, but notice how this kidney has an indentation right here? right? Right here is an indentation. It's drawn
Oh,
like a
okay.
bean, right? This is an indented bit. And you notice all the tubes, ducts, blood vessels are coming in and out of that indentation. That's what you call a hilus.
Hilus.
Hilum, hilus,
Where's
hyla.
that?
It's plural.
Oh, that's
But I don't ask it because there's no way I can stick a pin in there because you won't know what I'm pointing at. But it's an indentation in an organ with ducts going out. Lungs have one, lymph nodes have one, kidney has one. It's one of those terms you probably know what it means, but I'm probably not going to ask it. I could ask it just this way. The indentation in an organ where the ducts go in and out is called
Hilus.
That's all. So the blood vessels, kidney model. I don't want to ask the blood vessels on the preserved kidneys short of the renal artery.
I have
That
a
I can ask
fire
or
drill.
anything else because we already know the renal artery. But all the anything else, pretty much
They
exclusively
just throw them all
on
in the crib
this
and take
model.
them outside.
Are you for real?
My baby's having his first fire drill at daycare today. They throw all the babies in the crib apparently and just roll them out.
Oh my god.
Put 'em in the dumpster.
I guess it's easier than
They're
having to
trying
like
to
run
carry
and collect all
each
6
one.
kids in one arm. Like,
they're just all in the crib
I got the
together.
kidneys. I don't care about the children.
Wonderful.
Oh, so you got what's on there. Now the only other thing I should do today while we were here, I've already posted the
Oh,
slides
it's a
up.
lot.
But that's it. That's it for the renal system. That's it for everything you need to know in lab. I need to go over a couple of slides.
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 Are you
No.
They're posted on
serious?
Blackboard, but you got a bunch of slides. Not counting the white blood cell slide.
Oh my god.
Let me see if I can find them. They're too high magnification, so you can see the
Oh
stuff you've
yeah,
got
so
in
take a
here.
picture of that. I've never seen it.
You didn't draw it?
I did,
I'm
but
just
then I drew it too small. Look, I drew it
I
too
can't
small.
draw,
Hold on,
so-
hold on,
Crap,
hold
I'm
on.
erasing. I need the screen.
I need a picture. Okay.
They gotta be an AMP1, huh?
Mm-hmm.
All right. This is actually from material that was on the first lab exam. I just don't give you the slide because the first lab exam has enough junk on it already. This is simply a slide on an artery and a vein cross section. Vein, artery. You can tell the difference. This vein, look how big it is. The diameter of the lumen is huge because veins are bigger. It's also all collapse looking, which is typical of veins. They're so thin walled in preps like this, if there's nothing, no pressure on them, they just collapse. This is an artery. Even though there's no blood in it, all that, massive tissue in the wall tends to make it keep its shape. So artery, vein, that's about all you need at this point. I'm going to zoom in on the artery because those tunics, you can see those pretty easy on an artery. So here's the lumen where the blood would be. You see this dark line lining the inside surface there? That's all you can see of the tunica intima. More or less the base of the membrane. You can't see much of anything else. It looks like a line. Then you've got this big, massive layer. That's the tunica media. This is smooth muscle. You see this black wiggly stuff? That's elastic tissue. On histology slides, typically, if something is stained black and you don't know what it is, your guess should be elastic tissue. There's an iron-based dye they use to make elastic tissue stand out. It's almost always bluish black like this, so that's elastic tissue. So that's the tunica media.
Bless
Quite
you.
thick. And then the tunica externa is just this thin layer of connective tissue out here.
Bless you.
I've got it labeled as adventitia on this slide. It is tunica externa. That's an alternate name you'll see in some text. So those are the 3 layers. Tunica intima, tunica media, the thickest, and tunica externa on the outside. That's it for your blood vessel. I might ask you, if I gave you this slide, is this an artery or a vein? Say vein. Is this an artery or vein? You say artery. If I went here, I might ask you, identify what is this? You'd say an artery or a blood vessel. And I'd say identify layer, or identify layer, or identify layer. That's about all I get. Now this next slide. This is a slide of the lung. I'll look at that one. It's a common example of simple squamous epithelium. But what you notice in here is there's mostly nothing there. It kind of looks like a spider web.
Is that adipose? No,
Nope.
it's not. It's
This would
alveoli.
be air. This is air. This is air. These are the alveoli. Those air-filled sacs of the lungs. So this is the wall of the alveolus. This is actually simple squamous epithelial blood vessels in here. That's part of the respiratory membrane. There's air with oxygen here and here. There's actually some blood vessels running through there. So that is just a high magnification of the alveoli of the lung. These are just air spaces. Nothing can be there. The next slide is a slightly lower magnification of the same thing. See all this spiderwebby looking junk up at the top? Same thing, those are spider webs. Those are alveoli. They look like spider webs, they're not spider webs. So that's all air, and that would be simple squamous epithelia. Now this thing right here, this is an airway. How do you know it's an airway and not a blood vessel? See those yellowish looking lumps right there? That's hyaline carbon. You don't find that in blood vessels. That's a bronchi or a bronchiole, one of the airways. So this is a low magnification through the lung. So all you have are alveoli, and you got an airway called a bronchi.
So this is going to be on what, the next lab exam?
Last lab exam has a few slides on it. Most of them are white blood cell slides, but a few of these. Then, we've got like one or two from each of the organ systems. This one is the trachea. Remember the trachea? This one is just showing you the thick C-shaped hyaline cartilage ring. This is a lumen, air would be here. That's supposed to be the ciliated epithelial. It's not a great slide of that, but you got a big C-shaped hyaline cartilage ring. Oh. All right, next we got some digestive system slides. There's about 3 or 4 of them, I think. This is a section through the esophagus. Now, we haven't gotten to the digestive system yet in lecture. We'll get to it this week. But the alimentary canal, which is a tube that starts at your mouth, ends at your anus. The walls of the alimentary canal are also made up of layers. You can see them really easily on this diagram. See this dark layer right here? That's called the mucosa. You can find it in the esophagus, stomach, intestines, the works. So mucosa is almost always more darkly stained. Then you see this lighter area underneath it, kind of runs along here and up here. It's called the submucosa. It's basically loose connective tissue. It always stays pretty light. And then the third layer you can see on this is smooth muscle. There's muscle lining the entire alimentary canal, so pushing the food along, pushing the feces out, making everything move. There's an outer layer, but that's just the serous layer. The visceral peritoneum would be out here. It's not on this slide. Mucosa, submucosa, muscularis. Let me show you on the next slide, because this is also an alimentary canal. This is a section of an intestine, but you see the mucosa here, dark, then this is a submucosa, and you see 2 layers of smooth muscle there. That's the muscularis externa. This is typical of the entire alimentary canal. These layers are pretty easy to spot. Innermost one is to be where the food or feces is. Mucosa, submucosa, muscularis externa. The only other thing I'll mention on this slide, you remember on your lab sheet it had villi on there a lot, and I said you can see them in the cat, but I can't point to them. These are the villi. That's why I said they're easier to see on a microscope slide. This is the inner lining of the small intestine. It's got all these little finger-like projections sticking out to increase surface area. So those are the villi. They'll be along the whole small intestine. Now, another thing to note is the alimentary canal is one long tube, about 30 feet long, from your mouth to your anus if you stretch it all out. But there are different regions. Like in lab, we look at the esophagus, right? We look at the stomach, we look at the intestine. Well, how do you tell which is which? This is the esophagus. So tell about what it's going to have those 3 distinct layers you can see. But look at the mucosa. Compare this one with this one. See how the mucosa is quite different? The esophagus really doesn't do anything. It's a conduit, right? The esophagus takes your food, pops it down to your stomach. It doesn't do anything, doesn't digest, nothing. And your food, when you just eat it, think of crackers and things, they're kind of harsh and sharp-edged. Mm-hmm. So the esophagus is lined, just like the outside of your skin, with stratified squamous epithelium, so that withstand that abrasion. So really, if you look at the mucosa, that gives you an idea of what part of the alimentary canal you're in. This is stratified squamous. It's not intestines, it's not the stomach, it has to be the esophagus. When you drop down to the small intestines, you can see the mucosa, but it's got all these little finger-like projections. That's small intestine. It's got the, and if you zoomed in on it, they're not stratified. Small intestine has just single layer of columnar epithelia in it. It's not multiple layers itself. Microscopically, we're not going to tell it duodenum, jejunum, ileum. This is real small intestine. This is one you can ignore. It's a very high magnification image of the kidney. You don't need it. I think on the videos I'll cross it out. Same thing here, you do not need this one. In fact, I'm going to skip this one. Well, I'll talk about it here quickly.
We don't need that one?
Well, there's a bunch of liver slides. Liver tissue itself doesn't look like much if you haven't looked at many slides. I mean, this is actually the liver. Can you see anything in there? Well, no, I mean, I see nuclei. There's red blood cells in there. What happens in the liver? You remember you have that hepatic portal vein, brings the blood back into the liver, then it splits into another capillary bed. Well, the capillaries in the liver are referred to as sinusoids. They're really big, really leaky walled capillaries that form all these spaces between the liver tissue. This is where the liver detoxifies things, pulls out the carbohydrates and lipids. You can't tell anything from this picture, but if I gave you any liver picture, it would just be like this one. You remember in the liver you have those bile ducts, right? You also have blood vessels running through the liver, branch through the portal vein and hepatic artery. If you scan around liver tissue, most of it looks like this out here. These are just blood sinusoids with liver cells all around them, hepatocytes. This area, you might see it described as a triad in a textbook or something. You see a tube here, tube here, tube here, see a bunch of tubes. See this big one right here? You can actually see blood cells in it, so this is definitely a blood vessel. This is a branch of the hepatic portal vein. It's a really big vein. See this little tiny one here with kind of thicker walls? That's a branch of the hepatic artery. And then you see this one here? The cells lining this duct or tube are columnar. It's not a blood vessel. This is one of the bile ducts that come out into the hepatic duct, cystic duct. And you can often tell because the cells often are stained with pigment. They're much darker. But that is a slide of the liver. I'm less likely to use this one than the others. And I won't use any of the high power magnification ones. This one, don't worry about. All right, this is the only kidney cell I will give you. It's easy. This is a high magnification image of the cortex. What you are seeing here, this is a kind of oval-shaped lump of capillaries. This is the glomerulus. Then guess what this white space around it would be?
The capsule.
That's the Bowman's capsule. That's the lumen of the Bowman's capsule. So this is where material filters out of the blood and it squirts into the space here. Now, because it's a section, you can't see what it's connected to, but this would be connected to the proximal convoluted tubule and loop ahead and on down the line. So all I can ask you is, glomerulus, Bowman's capsule, this is out of the kidney. That's it. Don't worry about the high power magnification images of the kidney, like, well, I think this one's pretty. I don't think I'm going to use it either, though. This is one of the salivary glands we'll look at. Let's see what we looked at in the lab. The parotid. These are the ducts of the salivary glands. They're just made out of these striking columnar epithelial cells, and the nuclei are nice. They stand out a lot. But that's why I'm not going to. This one, I'll give you one stomach slide. The stomach is part of the alimentary canal as well. You remember what they call those internal recording-like folds on the inside of the stomach? The rugae? Rugae a little bit. That's what you're seeing here. This is one fold. This is one rugus. So you're not seeing all three layers. This is part of the mucosa. This is submucosa, and you can't even see muscularis on this slide. But what I wanted to point out, we'll talk about it a bit in lecture. The stomach is secretory, right? You know it secretes stuff, right? Gastric juice, it has hydrochloric acid in it and other things. See these little holes coming out here? These are the gastric glands that open out through what are called gastric pits. These are producing the gastric secretions, the hydrochloric acid, the mucus, the protein digesting enzymes. So these are gastric glands. This is where the food would be. You got mucosa and a little bit of submucosa. That's it. That's the stomach. There's some higher magnification images of the stomach. I won't use them, but you're getting pretty high magnification here at one of the gastric glands. This is really zooming in. I'll just tell you why they're on here. If you have the time to look at the histology, you can look at these gastric glands and you can tell which cells produce mucus, one of the stomach secretions, which ones produce hydrochloric acid, and which ones produce enzymes. They stain a little bit differently. You can ignore them all. We don't go into that much depth in here. But, you know, like these cells here, you can see that granular sort of cytoplasm in them. I think they're protein-producing cells, some of the protein digesting enzymes. So that's why there's high magnification ones in here. You know, here, these are the cells that produce mucus in the gland, so I don't use those. I won't do these either. I think these produce the hydrochloric acid. Oh, this is my favorite one. What's this? Yay! This is just a section through a tooth. You got the pulp. Oh, damn. Well, plus for reading. You got the tooth.
Well, and listening.
That's the pulp cavity. This brown looking, sort of striated looking stuff, that's dentin or ivory. That's like the bone. And then this amorphous looking brown stuff on the outside is the enamel. So yeah, it's just a cross section through a tooth.
That's kind of cool. Wait, so do we have to know that?
They do. Yeah. Come on, that one's easy. Got 3 parts to it. And it's very distinctive looking.
That's three
So here, I'll go back to
actions.
the beginning, right? So you need this one. There's the layers here. The lung with some airways, possibly. Trachea, yeah. This should know the layers, mucosa, submucosa, muscularis externa, and it's the esophagus. Intestine, again, the same 3 layers. No, 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 maybe, no, yes, that's a glomerulus and Bowman's capsule, and no. Stomach, yes, no, 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 yes.
No, yes.
The video that's out, it's just this PowerPoint slide. I think I narrated. I think that's what I posted. Is it narrated? I think I'll tell you. I don't ask this one. I don't ask this one. It'll do the same thing. It'll just cross them out. Say, don't worry about that one. So there's only like, what, 5 things on here. It's not much. So the only slides on the live exam you have 1 week from today. Are you excited about that, Gail?
I am.
Woohoo! You will have slides on white blood cell identification. Probably 4 or 5 of the scopes will be white blood cells. It'll be identify the blood cell, give me its relative frequency, and a 1 or 2 word function, just like we did on a quiz. The remaining 2 or 3 scopes will be off of these slides. You know, a tooth, an intestine, a stomach, a kidney.
Wait, so for the blood cells, we'll do like the percent and like what it does.
Right, what is this? You'll say neutrophil. What's its relative frequency? 60%. What's its function? Acute infections, phagocytic. So 5 scopes, 3 questions each, that's 15 questions on white blood cells.
Or when you're gonna write.
I could ask a red blood cell on platelet as well. Like I said, if you can't get a red blood cell, you know, you don't need to do it anymore. So that'll be about, what did I say, 15 or so questions. Then you'll have respiratory. You know, shoot for about 25. Then we'll have digestive. Shoot for about 25. We'll have renal. That'll be a stretch to get to 25. It'll barely make 100 questions. In fact, that's only 90 right there. So it's not as, won't be as long because there just isn't that much on it. So all the slides, 5 out of the 8 scopes will be white blood cells. The other 3 will be some of these. You don't know which one, but.
How many slides is in total, you said?
Pardon?
How many slides will it be total?
There will be 8 microscopes, 5 of which will be white blood cells. 3 will be whatever's on that PowerPoint. Then roughly, you know, it might be 30, 30, 30, but you know, about 25 respiratory, models, cats, you know, 25 digestive, models, cats, 25 renal, models, big kidneys. And that's it. You have a week. Now what we'll do on Wednesday, I'm gonna get together with Marsha Bowles, who teaches